Okay. Bye-bye, Lilia.
Bye-bye. It was nice to see you.
Bye.
Sí, sí, por eso. Now I can take this off.
Okay, great.
I am fine there.
Yes, yes,
Higher.
yes.
I'm fine like that.
Yes, yes, great, great.
Okay, this is good morning, or is it?
Could you just one moment,
what?
Enrique? Just one second. 1 second. Okay.
Sergey.
Just 1 second, because okay, so we just 1 second. I just want to okay, I will turn off my sound.
Okay.
Okay, so you can start now.
Okay, so I wanted to know, should I say good morning, or is it going to be in the afternoon, or when is it going to be? Wait. I can
Okay.
talk to me.
Yes.
Am I doing the video now? Was I recording the video or not?
Yes, I'm recording the video now. You should not record the video. I do it for you.
Okay.
Okay. Just turn on your camera. Because otherwise.
Because let me do something here.
Okay, no problem.
So I'm gonna.
Okay.
Because I don't see where I start here with the recording. Wait a minute now. Okay, here. Now I will.
Oh, okay, great.
Good morning, ladies and gentlemen. It's for me a great pleasure to be here today presenting the talk entitled Allergen Immunotherapy, Past, Present, and Future. It also has a subtitle of From Leonard Noon to New Vaccines Targeting Dendritic Cells. No, it does not work. It's not moving. Wait.
Oh my God, it's a little bit troublesome.
Yes, because it is not, have to, okay, now I have it. Now
Okay.
I have it. Okay.
Great. Okay,
Because
let's start.
It's not working with the pointer. I have to do it with my the mouse. So I will do it that way. Okay.
Okay. Yeah.
Okay. It is for me a great pleasure to participate.
Oh, Enrique, sorry. Turn on your camera.
You see, that's what I. Okay, now.
Okay. Yes, I can see you now.
Okay. It is for me a great pleasure to participate in this conference with the presentation entitled Allergen Immunotherapy, Past, Present, and Future. Allergic diseases are the most common chronic diseases in developed countries. The prevalence appears to be lower in developing countries, but however, there is evidence that there is a sustained increase in its prevalence. There are various age-dependent and organ-specific manifestations that contribute to the global increase in allergy. So you can see this, that under the umbrella of allergy, you have different conditions such as asthma, dermatitis, rhinoconjunctivitis, anaphylaxis, food allergy, urticaria, drug allergy, and other chronic inflammatory disorders, which also increases the total number of patients with allergic conditions. It's now clear that hundreds of millions of people in the world suffer from rhinitis, and an estimated 235 million people suffer from asthma. Allergic symptoms have a negative impact on the life of an economy of patients, and this has been seen in many countries and in many studies. Allergen avoidance and drug therapy by themselves alone cannot control the disease, and therefore only allergen immunotherapy has disease-modifying potential. Immunotherapy for allergies should be included in the optimal treatment strategies at all the levels and in all the countries. In recent years, there have been increase in the knowledge of the allergen extracts, but we all know that the composition is very complex. It contains allergic and non-allergic proteins and molecules and glycoproteins, carbohydrates, major allergens, minor allergens, pan-allergens, which are present in different allergen sources like tropamines, tropomyosins, profilins, arginine kinase, and so on. And these allergens can have different characteristics that can affect each other, such as cross-reactivity or proteolytic activity. These are biological products and as such are subject to a great variability. But however, when people inhale these allergens, you cannot differentiate, and in the end, you would end up inhaling different pollens and different molecules from different species. The natural history of allergy is a clear tendency to worsening the symptoms, polysensitization, a tendency to polysensitization, and association of rhinitis and asthma. You may start maybe with rhinitis and end up with rhinitis and asthma. Polysensitization is also very common, and it begins in childhood, and it is rare to find monosensitized adult patients who debuted in early childhood. There is an additive effect that promotes and increases inflammation and favors future sensitization once the patient is already sensitized. So therefore, the cross-reactivity, it's a therapeutic challenge to decide what you are really going to use in the vaccine, but also polysensitization is a diagnostic challenge to know really what the main cause of the problem is. And so this is why it is important to early intervene, early in the disease process. So the early diagnosis, what can we achieve? Well, the etiological diagnosis of the symptoms is ensured. We know early on what happens. Early specific diagnosis is important for the success of a treatment such as immunotherapy, because over the time, the patient acquires new sensitization, which complicates the selection of the allergens in the vaccine, but it can also reduce the chances of successful of the treatment. The evolution of the disease is avoided by administering the necessary allergy immunotherapy and treatment, avoiding the excessive use of symptomatic drugs. So if you start with the immunotherapy early on, you will also reduce the use of symptomatic drugs. We have to remember that most of these allergic diseases, they have different routes of exposure, such as inhalation, injection, or skin injection, or skin contact, and these will, most cases, result in systemic symptoms such as conjunctivitis, rhinitis, asthma, urticaria, atopic dermatitis, angioedema, or anaphylaxis, which is the most severe. So we have to consider all these in this conjunction. But it's important to know that the natural history of allergic diseases goes from allergic rhinitis to bronchial hyperreactivity or asthma, or from monosensitization to polysensitization. And that the only way that we can revert that is by using allergen immunotherapy, not by using drugs, and not, we don't have it clear also with biological products. So allergen immunotherapy would be the only one right now to revert this process. The early history, we all are aware of the early studies of Bostock and Dunbar, and then the first studies of with the prophylactic inoculation against hay fever by Dr. Noon, and then Dr. Noon and Freeman, and then they did the first studies, and they did the study in the UK with grass pollen, and they also saw, they also standardized the units in the noon units, and then they saw that they would have clinical benefits, and they observed a series of things that would happen during the treatment. So we know that it has been used empirically for over 100 years now, so it's one of the oldest treatments that we currently have. And it has been also accompanied by advances in the immunological field. So we know much better the mechanism of action, efficacy, the allergens, mediators, or the immune response. But also it has followed the normal evolution of a pharmaceutical product, which is increase the efficacy, reduce adverse reactions. The serious systemic reactions are the greatest concern, but we now they are easier to use, they are safer, and there are also different protocols to shorten the immunotherapy process, which consists in rush, cluster, or different schedules. Here we have a slide illustrating the different the evolution of the process. The first study paper published in 1911. Then you can see the use of aluminum hydroxide in the 1930s, which started in the US. Then in the 70s, you have the first allergoids that were introduced. But earlier in the 60s, you had the discovery of the mites in the house dust, also identification of the IgE, and that was an important thing, the first mite vaccines. Then you realize that allergology as such a specialty was done in Spain at the end of 1970, about 1978 started the specialty. And then we have at the end of the 90s, we have the sublingual immunotherapy, and now we have also the food immunotherapy. So some of the things have been not reached the market yet, such as the new recombinant allergens and CpG motifs, DNA conjugated allergens, or haptenic and low molecular weight products. So right now we have the native, the allergens, and sublingual, and some food immunotherapy products. This is a newer cartoon. You see here the first randomized double-blind placebo-controlled studies in the 50s, then IgE discovery, which is important for the specialty. Venom immunotherapy is also important in the 80s, allergoid, sublingual, a better understanding of the mechanism, some liposome adjuvants, and the WHO position paper, which was very important for the specialty. Then from 20 to 2014, then some studies with recombinants, peptides, anti-IgE combined with allergen immunotherapy, food allergy, intralymphatic, epicutaneous, sublingual, important guidelines and position papers on sublingual, and the EACI guidelines also in 2018. And also everything is aiming to precision allergen immunotherapy, which is also accompanied by important advances in the recombinant technology and biotechnology. But you can see also here how all this understanding of the allergen mechanisms have had an evolution throughout the years, from the early studies, from Noon, first immunotherapy study, all that people would talk about reaging, which was the toxin in these products, in the pollen and so on. And that was before the discovery of IgE. Then the mechanism would talk about a switch from IgE to IgG4, and this also lasted several years. Then we add to the paradigm also the Th2, Th1 differentiation, being more of Th2, more of the allergic patients. And now we're talking about Tregs, T regulatory cells. So you can see how this field has also undergone an important evolution. But also there's been an evolution in the products that have been used. We started with the aqueous extracts, then with that products with aluminum hydroxide, the allergoids, sublingual, some new adjuvants, tablets, and now allergoids with mannan, and some in gray here, which are not quite ready in the market, like recombinant allergens, recombinant allergens with adjuvants, intralymphatic immunotherapy, peptides, and so on, which are still, even the intralymphatic, we'll talk about it later, is used in some countries, but it's not a common, such a common practice. Things that we can conclude after 100 years is that it is clinically effective, that it is allergen or antigen specific, and it had demonstrated mechanisms of action. So important additional effects which are not shared by drug treatment include a long-lasting effect after 3 to 2 years of after stopping the treatment, that it prevents new sensitizations, although more studies are clearly necessary and needed. Reduces the risk of asthma in patients with allergic rhinitis, and then there are some prospective studies showing that. And also these effects have pharmacoeconomic implications to establish the place where immunotherapy should clearly be situated in health services in different countries, and there should be some consensus about all that. This is an interesting study which also should be repeated, and it is that in children whose parents had allergen-specific immunotherapy for 24 months, there was a reduction in sensitivities in these children. In other words, if the parents, especially the mother or the father, especially the mother, had undergone specific immunotherapy, the risk for the children of becoming allergic would be less. These were the group before, and you can see that there are no significant differences in group A or in group B. And when you did the study in these children, you will see that there was a significant reduction in positive skin creep tests in the number of children. There was also specific IgE values, polysensitization, early childhood bronchial asthma, wheezing, allergic rhinitis, atopic dermatitis, and total serum IgE. So in other words, these children had less of an allergic phenotypes if those who were born from the parents receiving immunotherapy. Of course, this study should be repeated and evaluated in different countries. Another thing that is also a growing debate is that absolute or relative contraindications to allergen immunotherapy. And this can be seen in this slide by different allergy societies of academies. As you can see, what is a absolute contradiction could be relative in other places, which are relative could be absolute in different places. So there is a greater concern about this, but this has been now, now they have absolute contradictions or in absolute contradiction or so on. So there is relative or absolute possibilities. And that now this has been also presented in the pediatric allergy immunology from 2020, and then where you have general indications and then general contraindications. And this is like a summary of all the other contraindications that we saw in the previous slides. And here is, you can look at the reference and see what is contemplated in this paper. So, but where do we stand right now? So let's talk about the past. The past, greater concern about efficacy than side effects, empirical use, unknown mechanisms, durations, and those were not determined, little knowledge about the allergen triggers, the house dust mix was used, house dust, mixture of multiple allergens, and so on. The schemes were slow concentration increases, endless treatments, mostly aqueous native extracts, mixture of multiple allergens, mixtures with allergens that inactivate each other, like mite and pollens, or with molds. The doses were really low to prevent these reactions. If not, they would get more reactions. And they were aqueous and then standardized in immune units, BNU, weight by volume, material of uncontrolled origin, variability between batches, and so on. And also, there was also common practice that the allergists would prepare and dilute their own extras in the office. And this was true until the mid-80s in Europe, and to a certain extent is still true in the US. However, in Europe, in the early mid-1980s, regulations were established regulating the manufacture and packaging of allergenic products. And they had to be prepared and manufactured under good manufacturing practices in the pharmaceutical industry. And furthermore, pre-skin tests are also considered drugs or pharmaceuticals. In the present time, and I put from the 90s to 2020, these extracts are medicines, biological standardization, GMP. They have in the pharmacopoeia, they need registrations. There are many documents that support its efficacy and safety, such as meta-analysis. There are new ways of administration, especially skin, subcutaneous, and slit, and also accelerated skin rash or cluster dosage. And this is also, the doses are better described, optimal maintenance doses, and so on. Also, allergoids are widely used. And also important thing is that the molecular diagnosis is also here, and there's some work that would facilitate the choosing the correct formulation. And there were some first studies with the recombinant starting in the year 2000. But I'm going to show you, sorry that this slide is in Spanish, but it is easily understandable. You can see here the concentration and then the time you need for up to 12 weeks to get to the maintenance dose. So you see that the induction phase is very long. With the cluster immunotherapy, you can see that we can shorten this time considerably, and the patient will reach to a maintenance dose much earlier. And using cluster, that could also vary. And then by using a rush or a super cluster immunotherapy schedule, you can see that in 1 or 2 weeks, the patient is already in the maintenance dose, and you could probably see a clinical improvement much earlier. So this is also something that has been significant and has improved the treatment with the allergen immunotherapy considerably. So this is it's a cluster pattern that we use in a paper that we published a few years ago. And with grasses, you can see the first day, two doses, and then 1 week later, 2 doses, then 15 days, and then monthly injections. So this is a very much easier program than going by weekly increments, and it would last forever to get to the maintenance dose. So now there was also a debate between SCIT and SLIT. And then, well, now we conclude that clinical efficacy of SLIT is comparable to SCIT. SLIT is more secure than SCIT, but when you compare SCIT subcutaneous with native allergen extracts, where the safety is not as good as with allergoids. There are some aspects that should be confirmed with SLIT, so like the optimal doses, the treatment duration, long-term efficacy, preventive action for the development of a new allergic manifestation, mechanism of action. So as you remember from the beginning, sublingual immunotherapy had not had so many years of use as what happened with SCIT, and there is less information available. There is some published meta-analysis comparing SLIT and SCIT, and you can see here SCIT, SLIT, 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 SCIT, SLIT, SLIT, and then the results all show that there is a tendency for improvement in both types of treatment. And then this was regarding effect on symptoms, and this is regarding the effect on medication requirement. And you can see that the efficacies are quite similar. Now, to what Immunotech has available, we have native allergens and sublingual way, and this is a rapid treatment, maximum dose from the first day, and it has 1 or 2 vials and the concentrations, and this is used with this pump per lingually or sublingually, and then it shows a good clinical efficacy. And then in the field of the allergoids, we have these alzoid, we have different preparations, and then The schedule is also quite rapidly, and you can see you can see that the building up is very quickly, and then that we also show studies with good clinical efficacy. So to what we have nowadays with immunotherapy, we know that it has a good clinical efficacy, the manufacturing process is well controlled, it is safe, it is convenient for the patient, it could be applied for many allergens, inhalant, pollen, different pollen types, for foods, venom, molds, and so on. So they have a well-defined, animal danders and so on, they have a well-defined process. And then it's also suitable for prophylactic application. It has no sensitizing potential, but it could prevent, as have been seen, the immune sensitizations, and also from going from allergic rhinitis to asthma. There are some unmet needs, and this is always important to mention. So we have to better define these homologous groups, and homologous groups means grass pollens is a homologous group, tree pollens is a homologous group, mites is a homologous group, foods, dander, and so on. Then standardization of rare allergens, and the definition of rare allergens is an allergen to which less than 10% of the population is sensitized to. So that would be a rare allergen. Shorter duration of immunotherapy, evaluate the use of booster doses as with other vaccines, like every 6 months after finishing, or 1 year, or whatever. Multi-center studies with the new products in SCID and SLID. Multi-center pediatric studies, pediatric investigational plan, the PIP, to evaluate safety and efficacy, and at what age treatment can be started. As we saw earlier on, people say 5, some people say 2, and then it's not an absolute, it could be absolute contraindication in some countries and maybe not in others. The use in primary and secondary preventions, identification of biomarkers to select people who respond to treatment and evaluate efficacy with objective parameters, improvement in the security profile, harmonization and validation of clinical efficacy criteria in different countries and different settings, cost-benefit analysis, social, economic, and health policy considerations between countries, standardization of products between companies, and also the use of allergen immunotherapy for food allergies. For future, so when does the future start? We don't know if the future is in 1 minute, or if the future is in 10 minutes, or in 1 year. So, but let's say we have now new ways of administration, different routes, vestibular, intralymphatic, epicutaneous, intradermal, administration schemes, cluster and rush are every day gain more popularity. Then the selection of the allergens according to the molecular diagnosis of the patients, with the 3ID, component resolved diagnosis, the recombinant allergens start to be available, aggregates with new adjuvants, what we have, and fusion proteins, and so on. And also important other indications for such as allergic dermatitis, for food, oral allergy syndromes, other food-related syndromes, and this should also be further investigated. So, as we know, there have been great improvements, not only in diagnosis, but also in the treatment of immunotherapy, and we have different possibilities, and natural or modified extracts. Allergoids are firmly established as a solid way to go. Hypoallergens, hybrid allergens, allergen peptides, biologicals, small molecules. So there is a vast, there are many possibilities for the treatment, but also the possibilities for the diagnosis have also improved dramatically from the early years. And here are just some expensive examples, and the component resolved diagnosis, and so on. So different administration ways. These are all papers that are quite interesting and quite recent from 2020. And then you can see here the SLIT is firmly established and it has proven mechanism. Subcutaneous immunotherapy also Epicutaneous immunotherapy is now being used in some countries and for some allergies. Intralymphatic immunotherapy also started in Switzerland, but is also used in some other countries. Injection directly into the lymph nodes. And oral immunotherapy and swallowing is also used for some food allergens. So these are the main ways that we have routes of immunotherapy. This all, contains different possibilities and novel developments, modified allergens, especially allergoids, which are firmly established. Recombinant hypoallergens are in study, allergen peptides are in study, fusion proteins with immune modifiers are also in studies, and all these aiming at precision medicine with patient selection, phenotypes, endotype biomarkers, and also the component result diagnosis, basophil activation test, and so on. So this will be all contemplated in the future. About mechanisms, this is also a very interesting paper from 2018, where you can compare here slit and epicutaneous with subcutaneous, with intralymphatic, the mechanism of the IgE. So this is also an area where there has been a great improvement in the knowledge and understanding of this form of treatment. But what is there for us in the future? I mean, it has been said, the predicting is difficult, especially the future. And that what we ask, where are we going now with the allergy products? And we have seen already that we will have many different possibilities. And one of these possibilities that we are using in the company are these allergoids from purified allergenic extract, conjugation with mannose. And this is under the concept of targeting dendritic cells for an effective immunotherapy. We have the allergens and the mannose, and then we conjugate that, polymerize that with the glutaraldehyde, and then we have molecules, allergens, which are hypoallergenic, like the allergoids, but also they also do increase the antigen uptake by dendritic cells. This we published a few years ago, and this is by the basic scheme that we just saw, and then with the production targeting dendritic cells, they have an higher allergen uptake, and then the production of different mediators and the production of T regulatory cells. And we published that in 2016. And then we also got an editorial from the reviewers and from the group of the Paul Ehrlich Institute, and they also reproduced the cartoon with the different mechanisms and the mediators and so on. And this is also from a recent one from 2017, when they also reproduced this method of allergens coated with mannose as targeting dendritic cells. So this is a new approach that is gaining a lot of attention. And we have done now several clinical studies. So everybody thinks that the future will also be with recombinant allergens, but well, we have to think about this and see what are the pros and the cons. And then, well, theoretically, they should increase the safety of immunotherapy, modification of the tertiary structure of allergens, allergen oligomers, allergen hybrids of different molecules, allergen-directed mutagenesis, allergens conjugated to immunostimulating sequences, immunotherapy with recombinant allergens that replace a natural extract, or you can also complement or supplement the vaccine with recombinant proteins, immunotherapy with hypoallergenic variants of recombinant allergens, and also this could be also based on component result diagnosis. So if you see that the patient is sensitized to several recombinant allergens, with theoretically thinking about producing a vaccine with those recombinant molecules. Well, this started in the year 2000, and there have been many studies using recombinant molecules, especially from birch pollen, that the one, but also from grasses. There are also been from peanuts, and some other one from cat allergens. So there is a wide possibility of molecules which are available that have been purified and sequenced and cloned, and they're recombinant molecules that can be used from different settings in a theoretical way for doing these studies. So, but also, this sounds logical in theory. There is no difference between theory and practice, but in practice there is indeed. So, different legal requirements between native and recombinant allergens are much more severe and strict for recombinant molecules. Furthermore, what applies to natural extract does not apply to recombinants and vice versa. A recombinant allergen, one clinical study, one registration. Two recombinants, two clinical studies, two registration. Very high cost to bring them to market. And this is true of this approach of using recombinant molecules. And it has been seen, for example, as you can see up in the left, recombinant allergens, the main disadvantages always have been development costs. Hypoallergens, development costs. They all have, most of them have been tested in humans. But you can see peptide-based immunotherapy, development cost. And this is one of the problems associated with the launching and using recombinant, the main disadvantage of using these products. So, for ending my conference, allergen immunotherapy is the only option to establish a long-term medication-free cure of allergic diseases, and this would be ideally the goal. The use of modified allergens aims to increase, in this case, we're talking about the allergoids, increased efficacy and limitation of side effects such as risks of anaphylaxis, helps to better and longer presentation of the allergen peptides with no binding to IgE present in the patients, and this would be with the modified allergens. Then also precision medicine contributes to allergen immunotherapy by better characterization of patients, selection of custom-tailored therapy per patient, and monitorization of therapy success by biomarkers. So this would be what we want to see also as novel developments in the field of allergen immunotherapy. So when we develop a product for allergen immunotherapy, our main considerations in product development would be optimize the product profile for specific immunotherapy. For example, shorter dose, increased phase, simple schedules, flexibility when a dose is not given. Also regarding safety, reduce allergen activity, higher doses in shorter periods of time, same volume, and then compliance, less frequent shots, faster efficacy, longer efficacy or longer lasting effect, improved cost-benefit ratio, and reduce the use of symptomatic and reduce the cost, the overall cost of the treatment. And all these will end up in the concept of efficacy. So if we get all these and we think about these considerations, we will have a product which has simple use, safe, compliance, and is very efficacious. So to finish, I would say that talking about the past, well, okay, talking about the past, we should overcome the nostalgia for the present, then follow the path marked by scientific evidence in the present case, and also in the future, be aware of changes to provide more clinical benefits to your patients. And this was it. Thank you very much for your attention.
Okay, great, Christian. I have made the video. We will adapt it. Could you please send me the presentation which
yes.
show? Okay, yes, so we will translate it.
I will send you the presentation.
Okay, thank you very much.
It has some slides which are, some of slides are hidden, but you will not, they will not show up if you want to use it, okay?
Okay, okay.
Or I can delete it. I send you the final version.
Okay, okay, it would be great. It would be great. Okay, thank you very much.
Okay, what do you think? It was okay?
Yes, I think it was great.
Is it okay?
It was, yes, very interesting, very fun, great. Yes, thank you. I think that you know, your first video in Ukraine, your first lecture, just made a big success in Ukraine, because a lot it was just like fresh air for Ukrainian allergists. Because you know they usually talk about the same topics, and your lecture was very scientific, and they were just happy about it. I think that this lecture will have the same the same influence.
Okay, well, thank you so much.
Thank you, Enrique. Thank you very, very much for your time. So bye-bye.
I hope that I helped. Okay, thank you.
Thank
And I will
you.
send you the final slide in a few minutes.
Yes, yes. Thank you very much. Thank you.
Thank you.
Bye-bye.
Bye.
Bye.